Hey everyone, welcome. Zoltan Peristegi here from RotaChrom, and we're going to be starting now. I'll be your host for today's webinar, which is the third in our series. So if you're new around, you're welcome to listen to our first two webinars online. Our webinar will be partly building on information that we shared previously. So we'd like to encourage you to visit webinar.rotachrom.com and register there to view our previous webinars. Obviously, you're here now, so listen in and you can listen to the previous ones later. As you probably know by now, RotaChrom manufactures the world's first and only automated industrial-scale centrifugal partition chromatography instruments. Established in 2014 in Europe, RotaChrom has presence in North America, Asia, and Europe with applications in the pharmaceutical, natural extracts, food and beverages, and cosmetics and fragrances industries. The speaker of our webinar today will be Andras Gaspar, PhD, RotaChrom's newly appointed Chief Product Officer. If you have questions throughout today's sessions, you'll find a Q&A button at the bottom of your screen. We'll address as many as we can at the end of the session. We'll follow up one-on-one with every participant who asks questions that we won't have time to answer today. So you don't need to worry, we'll get back in touch with you. Andras, before handing over to you, let me first congratulate on your new position. Absolutely well deserved. And with that, the floor is yours.
Thank you very much, Zoltan, and I would like to also welcome you all for new participants and the ones who are already with us on the last two webinars. Today we're going to discuss, unlike in the last two episodes, we're going to rather on the economic side of this purification technique that we possess. The last two presentations, we were rather focusing on technical details, applications, scale up. And in this presentation and in the upcoming presentation, we're going to rather focusing on the total cost of ownership part, the CapEx and an OpEx side of this whole equation. So as mentioned before, I'd like to draw your attention to our next webinar, which is going to be somewhere in the middle of March, I guess also on the 11th of March, where we're going to present our total cost of ownership simulation tool that we developed over the last couple of months. But today we like to focus just a quick recap on the CPC. For someone is technology somewhat new. Just a quick overview. We like to compare this technology to technologies that are currently available on the market, and we're going to do this comparison via the cost structure of the ownership, and of course we're going to have a peek preview on the selection tool that we also developed for our customers. So without further ado, just a couple of words, and I'm not going into really technical details. We did in the last two episodes, so feel free to check them. But on this stage, at this time, we just only like to focus the approach and the differences between the two approaches, or the main differentiator of those approaches that currently on the market. The most common and traditional chromatography, that's also in the title, is a solid state support chromatography, where usually is better known as preparative HPLC or flash or supercritical chromatography, where usually the resolution, the way how it's the separation peak to peak resolution and separation of the individual components from impurities or isolation, usually happening via kinetic optimization. This involves the increasing the efficiency of the solid state, usually the silica-based solid state, particle size, pore size, distribution, and so on. While parallel, CPC is usually working from the selectivity point via a carefully selected solvent system, biphasic solvent system. CPC is able to deliver very same results and able to isolate or purify your compounds, but in a different way. Just what does this mean in a bit more detail? I just turn myself video off so you can see the complete picture. What does this mean? In normal preparative or flash chromatography world, where you have the solid stationary phase, you usually have a really high number of plates, and this allows you and delivers you a relatively high resolving power, and with that you are able to isolate compounds with a relatively high peak capacity. On the other hand, CPC, partition chromatography, has much lower on the resolution on the plate number side, but this is compensated with an extremely high selectivity values, which is not typical for preparative HPLC techniques. Also, the main difference between those two technologies is also coming from the solid state, where this robustness is somewhat deteriorated by the fact that you need to take care of your silica column. It's not something that you really want to risk clogging, or you really want to introduce material that is extremely heterogeneous. So on the other hand, CPC is relatively good for and robust for such complex matrices or material that needs to be purified. So just a quick summary from the last presentation as well. This way how we differentiate ourselves from traditional chromatography is also, and the way how the solvent-solvent biphasic system works in our system enables us to have a very quick and cost-efficient method development. The whole method development results in a solution that is easily scalable. So whatever works on our small machine or a benchtop machine can easily work on a larger scale machine, almost seamlessly transferred. For those who are aware of preparative HPLC method development, they completely understand what I'm talking about. It's a very, very tedious job to do something on the method development side on the traditional large-scale preparative chromatography. And also the advantage that we don't really possess a solid stationary phase, that this technology is rather forgiving. So whatever mistake happens during your method development or something comes in between during the cycles, separation cycles, excuse me, then in this case you can easily start over. There's not much harm done, and in that case you can start over without the loss of a significant chunk of your OPEX costs, for instance, the complete column. So in order to go in further detail with the cost side and not on the technology side, from the last presentation, you may remember that we analyzed the complete and went through the complete downstream process in this hemp industry, starting from the hemp biomass. And in that case, and starting from a dried biomass, going through extraction, winterization, decarboxylation, distillation, you end up a purification process to have a fine finished product or remediated product. And what we have not discussed last time, and we're going to start the discussion here, is the mass balance that's changing drastically over these steps, and the effect of the process cost and effect of the individual steps on your overall costs. So what you can see here is basically, it's relatively obvious. During the extraction method, the dry mass between the mass balance, the extraction and the dry mass is a huge change. It's a significant drop, and from that onwards is relatively lower, this loss. But parallel, it's irreversibly proportional the costs that you that may imply with that specific part of the technology. in that complete process. So maybe it's easier to see on the next slide that we had a customer. We also mentioned this case study in our earlier presentation, where the case customer approached us with a dried mass, and we went through the complete calculation of the entire process of theirs. So we're not only focusing on the purification step that we own, and we started to analyze the complete mass balance and parallel also the compound interest change, what you can see here. And of course, as I mentioned before, you have this significant drop of the complete mass. So you're starting from a ton of dried mass and you're ending up in around 18 kilos. It's a significant loss, but parallel, your compounded interest concentration is tenfold increased over this entire process. And what we like to emphasize here for us was also some kind of a shock, so to speak, that we analyzed all the costs of the individual steps, not only the capex but also the opex costs. So all these in the total cost of ownership calculation. We also involve the depreciation for the solvent handling and for the instrument itself for each step, for extraction, winterization, decarboxylation, and so on. And we realized that if we did our calculation, in that case, these initial steps are significantly lower than the last final step, chromatographic purification step. And this number, as you will see further on, might even be much higher in case of traditional chromatographic methods. But the main message here is that it's a well put effort to focus on each individual process step and optimize your complete process flow and mass flow in order to reduce the largest effect on your TCO, the final step, and carefully select that one at the end. So what does this mean? We came up, just because of this, as we started to discuss with different customers, we came up with this simulation method, where actually the total cost of ownership is, of course, comes together from many components like the depreciation, the consumables, the labor cost, overhead, all these related that are related to production cost. But parallel, this is not the only aspect that we need to investigate during an investment, but also there are additional consideration like safety or level of automatization, the uptime, or otherwise downtime that is necessary to maintain these instruments. What sorts of versatility this whole process flow brings you? We know and we understand that currently this market is extremely volatile. So it's a huge question mark that what sorts of product lines that you are producing. One day you are after remediated broad spectrum, other day you are rather looking into minors or THC remediation and so on. So these are things that needs to be also taken into account when an investment is ahead of you. So as I mentioned earlier, the major impact on your process and the total cost of ownership of your complete process is basically the purification step. And we're going to investigate this a bit further into details. And what you can see here is we did our homework in our method. We went into great details with the very same customer, and then we wanted to map out the exact cost structure of the output compound of interest. And as you can see here is relatively straightforward to see that the consumables, which are basically from our perspective, is the solvent, the solid stationary phase or any type of stationary phase, and the electricity that is required or energy that is required for solvent reclamation. That's the major part of your complete TCO. You may say that depreciation is the second most important part in this game. And of course, these main parameters are as I mentioned, the cost contributing factors within the purification section. So that's what we considered as a key KPIs for our benchmark that I'm going to show you in the next couple of slides. So these consumables, we think that these are the major cost drivers. We were focusing on these for the sake of technical comparison with the different commercially available techniques. We try to keep a fair comparison, and in each case, even most of the suppliers, they don't really do like we do. They don't really supply additional solvent recovery system. I believe RotaChrom is the single supplier to this space, where we also able to provide the tailor-made solvent recovery system for our chromatographic solution. And of course, to be again on the very fair side with our competitors, we also included solvent recovery. The level of solvent recovery was also identical. So what we took as a benchmark, as a case study, we wanted to have to figuring out what are the differentiators between our competitors, and of course during customer discussions to understand also from their perspective what are the major key drivers. So every time is usually comes down to the throughput requirement. For this purpose, we selected a single example, like 30 kilo a day, 24/7 operation with 90% uptime. And this is like a THC remediation case typical for us. Some of the assumptions that we did, we had a lot of case studies. We have well over 200 citations found for these list of competitive technologies that we found. We took some of the assumptions that actually not really relevant for us, such as the solid stationary phase cost, bulk cost, because we don't have those. But again, just wanted to be fair, so we took these efforts and did our homework and looked around and looked for all those solvents and solid stationary phase costs that are relevant for this comparison. So what are these technologies that we currently see on the market? And we've seen several presentations over the last year as well from our competition. And basic technologies that we have here on this slide is most of them are dominantly chromatographic-based technologies. So I'm not sure if you are familiar or not. Just wanted to really quickly go through this. The first two technologies are rather similar. Some of them is hydrodynamic, and the other is hydrostatic countercurrent chromatography, flash, prep, HPLC, simulated moving bag, or similar to simulated moving bag YMC, or the YMC product, YMC technology, or the supercritical chromatography. All of them are coming from the same background, using a solid stationary phase depending on what type, normal phase or reverse phase, and they forcing through some kind of solvent or eluent, and that's basically how the separation occurs, relatively similar to partition chromatography. Below the line, you see technologies, techniques, SOPs that are on the market as well, available. However, these are, I need to emphasize that these are mainly for THC purification, so THC remediation, while the other chromatographic technologies are available for many applications, and you can versatilely use those. These three technologies are basically only exclusively done and used for THC remediation. So these, just again, to be on the fair side, since this is a THC remediation case study and comparison, we also involve those, but some of them are really limited publicly available knowledge that they show or share, or some of them are even emerging technologies and they're not really at the stage the market penetration is not that significant like on other chromatographic technologies. So what our findings are, It's I wanted to point out, as I did at the really beginning, that in this comparison that we looked for technologies and suppliers and instrumentation of those suppliers that they have more or less the same throughput values, which is basically going to be the groundwork of our calculation later on the consumables. So in this slide, we just wanted to represent that more or less all of those technologies that are here in the same ballpark throughput range, except is the precision extraction SOP that you may see on the internet. But apart from that, these are the most, more or less the same. Then, of course, as I mentioned in the last couple of slides, the most important TCO driving factor is the total consumables, like solvent, electricity that's required for operation and solvent reclamation and the solid stationary phase. You will see that among these chromatographic-based technologies, RotaChrom is the only one that are able to break through this two-digit barrier, so to speak. So the total cost, the total consumables per kilogram remediated broad spectrum oil is lower than $100 US. Actually, in case of CPC, RotaChrom CPC is somewhat around 20-30 US per kilo. It really depends on the material, really depends on the input material. The separation method that we are able to apply is really heavily related on the input material as well. But more or less, we can say that those techniques, those emerging technologies are, and RotaChrom are the only one who are able to make that barrier, the consumable cost barrier. The next slide, as again I was keep pointing this solid stationary phase issue. We wanted to also to visualize what is the actual costs within the consumables that are really driving this whole story. And not surprisingly, if you're aware of chromatographic technologies, you see that the price and the costs of those bulk, even if it's coming in a bulk, the stationary phases are extreme. And again, to be fair against our competitors, we also signalize that although we don't have the solid stationary phase, but in our technology, we have an immobilized solvent as stationary phase, and the cost, we wanted to also put the cost implication here just for the sake of a fair comparison. And yes, this is something which is really visible, that in case of TCO, within the consumables, the stationary phase cost is your major issue or major problem during your OPEX, during your operation. And then we are more or less coming to an end, and this is something that here wanted to highlight, that we took a couple of months and couple of hundreds of publicly available data, white papers, publications, technically available technical details and sheets, what we found from our side, from the competitor side, and that's how we were able to come up with a truncated calculation method for the capex and opex values. And for those customers who are in contact, we always start with that sorts of benchmark analysis, because most of the customers are not aware of the hidden costs during the complete operation. So what you see in the right side, that with this small plot, that the cumulative costs that containing the initial capex and the opex cost over 2 years of period are significantly different between those technologies. So it's really depending on what you pick for your last process step as purification step, because some of those might look cheaper on the buy-in side. So some of the costs that you look at, the cost burden that you have at the beginning is really appealing, but over a couple of months you will see that their cumulative costs due to the cartridge, due to the bulk silica price and so on, it's going to rocketing towards a really high over accumulating costs. So this is what we usually do with our customers, and we go through together with on the competitive landscape to have a better understanding, and then let the customer decide what sorts of direction he is willing to take when they're going to invest in this complete process of hemp purification. So with that, I'd like to summarize what I was presenting in this short period. It's the very same. I left this statement here from our last presentation that partition chromatography is really flexible. We are able to change and adapt relatively efficiently, relatively quickly. So even within a day, you can change your complete product line or output product on our system, which is not really possible on other chromatographic methods. It's scalable, so scaling up your procedure from the benchtop to a large-scale, industry-scale CPC is seamless, almost seamless. It's efficient. The cost that we've shown you here, the cost structure, the total cost of ownership is for the complete process where the major driver is the purification step. And we are more or less aware of the fact that chromatographic method is currently the single one that you can go for. Among those chromatographic methods, CPC is one of the most efficient technology today on the market. And versatility, this is I already pointed out that at some stage where you are in this really volatile market, you are not really sure that or you cannot be sure that what's going to be your product line tomorrow and what's the highest value on the market today, or what's going to be the next target tomorrow. So, and another feature that is relatively unique on the market that we've seen via our study, this benchmark study, that the solvent reclamation, solvent regeneration system that we tailor-made to our chromatographic purification process is unique. Most of the time, you rather go out and need to solve this issue of how you reclaim or regenerate the solvent on your own. You need to find the proper supplier for that, independently of your purification supplier. But this is something that RotaChrom can offer at this stage, that we can bring it to your house at the very same time, optimized for your solution. So with that, I like to thank you for your attention. If you have any questions, then we are here to answer those. Thank you very much.
Thank you for today's presentation, Andras. I will now look through the questions. We have a bunch, so let me try to put them into order here, and we will answer them shortly, or Andras will answer them shortly. In the meanwhile, I would like to draw your attention to our upcoming webinar, which will be the last one featuring Andras for a while. So this is going to be, that's going to be your last chance to catch Andras speaking at a webinar. And that will focus on how to maximize your processing profits utilizing CPC. So we're going to dive a little deeper into the numbers. So I assume that many investors and business decision makers would be fired up by this topic. So if you are more on the technical chemical side, please encourage your business partners or investors to join our upcoming session. So you can just forward your invitation, or just direct them or send them an email with a link to webinar.rotachrom.com. And now let's see the questions. So first question is regarding the CPC instrument. So what is the average per day volumes of solvents used for one CPC?
Well, I'm guessing it's not going to surprise you. The answer, it depends. So it really depends on the method that you pick or the product line that you are after. Currently, let's stick with this case study that we presented here. For that kind of THC remediation study on the larger instrument, you may say that one run is around 20 minutes. During this run, you have around 2.5 liters per minute solvent rate that is going into the system and of course going out and needs to be reclaimed. So if you multiply this by an hour and you're managing 2 or 3 shifts, then it's relatively coming to a couple of hundreds of liters per hour or per shift. It's really depending on. We have situations where it's really high purity material is needed, or we have a smaller machine where this flow rate is around 200 mil per minute. So it's really depending on your procedure that you pick or your product that you are after.
Yeah, and if I may go a little further, I would just encourage you, the listener who asked this question, to get in touch with us, and we can give you very specific answers for your specific needs or your specific processes. Next question is about THCA remediation. If we at RotaChrom already have a method for this, and whether the resolution is better or worse than for THC.
That's a very good question. If you checked our previous presentation, most of the cases, and this is how the cost stack look like and suggest to us, and that's why we try to always push the customers to having a distillated fine product in order to reduce the cost burden on your total cost of ownership for the entire process. So the less material that you enter in the purification step, independently if it's CPC or any type of chromatography method, it's going to be lower your final costs. So that's why this is, by the time most of the customers of ours are reaching this state, they don't really have the acidic form of THC. We started to have discussions with customers that they are actually after. This is also a similar story when you have delta 8, delta 9 THC separation. These are really fine tuning stuff where most of the time we ask the customer to develop, or actually we offer them to develop a method for those unique questions. For generic stuff from THC remediation, THC isolation, pesticide remediation, and all of these stuff that for us it's getting normal, we have the data, we have the library, so to speak. But these emerging questions that we have with delta 8, delta 9, THCA, this is somewhat, again, what we see on the market. I would say that at this stage, you need to get in contact with us, with our subject matter experts, and they're going to help you to assist with the technology and the method, the appropriate method.
Yeah, so I would just extend that, Andras, if you let me, by stating that RotaChrom actually has a research laboratory with many chemists. I don't exactly know the number, but more than 10 on our team, happy and willing to work with you on your specific purification needs. So you see on screen the email address and the phone number. So just get in touch with us, and yeah, we will definitely be doing our best to develop methods for each and every purification needs. Next question is probably going to be easy for you, Andras. What is the loading capacity of the compounds of interest?
Well, again, it depends on. So if you have a really bulky, huge ballast containing crude material with a lot of green stuff, flavonoids, terpenes, and chlorophyll, and all of these things, and you still want to go for a remediation, for the large machine, it's going to have like 0.5 kilo or per run that you need, then you are able to inject because of the huge volume of ballast material or matrix material that may affect, you might understand, some of you that or heard about this matrix effect. But if it's a nice distillate, that most of the time we encourage our customers to work with distillates or mother liquor, then you can really push the boundaries. So you can really go around a kilo of solved material with a really high concentration. The generic starting material concentration that we have is around 0.4, 0.5 gram per liter. Is this correct? Yes. And then you can really push this with more than a liter of volume that you're injecting. Some of those, and again, it's the other side of the question is that what is your goal? If it's like a targeted isolation issue, then you really need to be careful how you overload the system. Or if it's a non-targeted, like a bulk from full spectrum to broad spectrum kind of game, that you need to chop off some part of your compounds and you need to remediate, then this is also going to influence. So the way the method that you are after is going to influence this story. Sometimes you can really overload. If it's like a non-targeted approach, you can really overload these CPCs, unlike traditional chromatography, and you can really push it these cycles. And some of the cases when it's about isolation, high purity, just the way how chromatography works, you really need to figure out and coming down from this overloading situation to a point where the compound of interest are not overlapping. But it's relatively easy to, in case of, just to give you without having too much blah blah, just to give you an idea, if it's like a 3 shifts it's a 3 shifts operation that we have, that's what you see. That's around 30 kilo of distillate could be pushed through the system, the larger system, per day as input material.
Thank you, Andras. We want to be taking your time into consideration. So actually, we want to allow 2 more questions, which we already have. But if you have questions, still feel free to ask, and we will get back to you directly. So we have a question regarding purification of cannabinoids to pharmaceutical grade. What scale can CPC achieve?
Yeah, so this is the question marks that we're getting more and more frequently. And the answer is usually for that it's really depending on the concentration or the state of the input material. If you check our previous presentation, this is something that we also encourage most of the case, even changing the complete procedure. That's going to be a much more efficient way towards mother liquor approach. But the isolation and of course this is a situation where you really need high purification, high level of purification, and the isolation is your main key focus. Sample preparation, you do a distillation or even you crystallization before, and then you're focusing on minors. You removed via crystallization the large bulk CBD from these distillates, and then you are able to concentrate your input material on the minor side. And then if you are after isolation, usually it's always a sacrifice on the throughput and the yield. But what we used to say that once you have a compound starting from a 50 to 50-60%, then with CPC with 1 run you can easily go above 90%. You can go to 99%. Depends on how much will you sacrifice from your material, because all those Gaussian type of shapes of curves and overlappings that you have from different materials, you need to cut away whatever is overlapping. So the higher your purity grade requirement, the higher the loss on yield that you have. But again, this is something which the beauty of chromatography, that you can achieve something that is really high level of isolation, high purity, and on parallel on the very same instrument you can achieve something that you are willing to compromise with a lower level of purity, but you will have a higher level of yield. So that's always a give or take. But yes, so to summarize up, yes, it is possible to push the purification towards 90 plus, 95 plus, 99% of compound. And again, this needs to be clarified at the beginning of your procedure. Are you going after isolation, or are you going after purification, so bulk kind of approach?
Thank you, Andras. Finally, our last question, actually, the listener is planning a full stack process chain, and is asking, comparably, how does my silica cost compare to RotaChrom's CPCs in case he'd like to remediate THC and he has 25 kilos per day distillate to purify? So very specific question.
Yeah, that's well, to be honest, we spent some time to able to answer that question. So the best example is one of a supplier. I don't really want to mention name. We have the technical data for that, so it's not something that we came up with. They have on their technical paper stating the level of costs that is implied by the solid stationary phase. So the best example for this would be a flash supplier who has an instrument which is relatively cheaper on the capex side, with the very same throughput capacity, so around 20-30 kilo a day, full 3 shifts operation procedure. In that case, they are able to process the same material that we do, so with the same level, but after 50 to 100 cycles, they need to remove and replace the cartridge. So if you're familiar with flash chromatography, you know that these are disposable cartridges. So the disposing of such cartridge is also considered to be hazardous waste. So it's not something that you put in a garbage bin. But the cost implication on that one, that you have 20 to 30 kilo of compounds purified a day, and let's say that it's almost the same level, so 20, 30, 40 minutes per run. You can calculate that how many runs you need for that volume. They state that after 50 to 100 cycles, a $90,000 price tagged cartridge needs to be replaced. So this would mean that after every 2 or 3 days, $90,000, $90,000, $90,000. So this is obviously not the situation with liquid chromatography, because you don't have that cartridge. You dispose and recycle the solvent that you used as stationary phase. So that would be the answer for that. It's a huge cost difference between CPC and flash. Or if you want to go into even more detail on the preparative HPL side, we can also do that. They are somewhat more efficient because they use bulk silica, and with dynamic axial compression, they are able to control the degradation of that column a bit more. So that price tag is not that heavy, but it's still at least an order of magnitude higher than with CPC.
Thank you, Andras, and thank you to all of our listeners for your time and attention. We truly appreciate it. I hope you found our session useful today. So if you're watching the live session, we will send you a link to view the replay of today's session shortly. One more thing, actually, if you would like to dig deeper into today's topic, you're welcome to join us on our monthly podcast called Purified, where I interview the speakers of our webinars and discuss the month's topic further. So search for Purified by RotaChrom on your choice of podcasts. Also, please do not hesitate to reach out to us at sales@rotachrom.com should you have any questions, or would be interested in learning more about our purification platform. Please stay safe during these days, and I hope to see you again on March 11th on our session on maximizing your profits. Don't forget to register now at webinar.rotachrom.com. Thank you for your attention again, and see you next time.
Thank you very much. Bye.